You know, out in the fields and hidden away in labs all over the world, there's a quiet revolution happening. Scientists are using these incredible genetic signposts to build better, stronger, and more resilient crops. Today, we're going to pull back the curtain and decode these powerful tools, molecular markers.
I mean, let's start with the big question, right? How are we going to feed everyone? We need crops that give us more food, that are packed with more nutrients, and that can handle everything from drought to pests. This is the challenge that keeps scientists up at night, pushing them to find faster, smarter ways to improve the plants we all rely on.
For centuries, we've done it the old-fashioned way, classical breeding. You find the best-looking plants, you cross them, and you hope for the best. And hey, it's gotten us pretty far, but it can be painfully slow. We're talking years, sometimes even decades, to get a new variety. And you're never quite sure what you're getting, genetically speaking. There had to be a better way to look under the hood, right into a plant's DNA.
So, here's our game plan. We'll start with the challenges breeders face. Then, we'll get into what these molecular markers actually are, and check out the different kinds. After that, we'll see how they're being used right now. And finally, we'll peek into the future of this amazing tech.
Okay, so how do you speed things up? How do you get past the limits of just looking at a plant? Well, you use molecular markers. Think of them as a GPS for a plant's DNA, little signposts on the genetic highway that guide you straight to the good stuff.
So what is a molecular marker, really? It's just a specific piece of DNA that we can easily find. The key is that this little piece of DNA is always inherited right alongside a gene we care about. So it's like a flag planted in the genome. If you can find that flag, you know you found the trait you're looking for, whether that's disease resistance or maybe sweeter fruit. But of course, not all markers are created equal. To be really useful, a marker has to tick a few boxes. It's got to be polymorphic, which is just a fancy way of saying there are different versions of it out there. Otherwise, you can't track anything. Ideally, it's codominant, which lets breeders know if a plant got a trait from one parent or both. And maybe most importantly, finding it has to be reliable, fast, and cheap enough to use on, thousands and thousands of samples.
This hunt for the perfect marker has led to this really fascinating story of evolving technology. So let's take a look at the major players in the game, from the old school classics to the super advanced tools we use today.
It all really kicked off back in the 70s with a technique called RFLP. It was brilliant, but man, was it slow. The real revolution came in the 90s when PCR, the DNA copying machine, hit the scene. Suddenly, there was this explosion of faster, more efficient markers like RAPIDs, SSRs, and SNPs. You could get results in a fraction of the time and cost. It was a total game changer for genetics.
To really get how this works, let's zoom in on one of the originals, RFLP. That's a mouthful. It stands for Restriction Fragment Length Polymorphism. This was one of the very first ways that scientists could actually see differences in the DNA code itself.
The way RFLP works is kind of like solving a genetic puzzle. You start with these special enzymes that act like tiny molecular scissors, and they cut the DNA at very specific spots. Then you separate all those little fragments by their size. And here's the cool part. Since every plant's DNA is a little bit different, the cutting pattern is totally unique. You end up with a distinct genetic fingerprint for each one.
Let's break that down real quick. First, you get the DNA out of the plant. Second, you use those restriction enzymes to chop it all up. Third, you run those fragments on a gel which separates them by size. And fourth, you use a special probe that lights up and sticks only to the marker you're looking for, making it visible. It's a few steps for sure, but it is super reliable.
So, as you can probably guess, there are trade-offs with these different marker types. RFLP, super reliable, but slow and a ton of work. RAPD is really fast, but sometimes the results are a little inconsistent. And this is where SSRs and especially SNPs just totally shine. They give you the best of both worlds. They're highly reproducible, and they're incredibly fast thanks to PCR. That combo is exactly why they're the gold standard in genetics today.
All right, enough with the theory and the tools. Let's get to the good part, the impact. How is all this technology actually being used to completely change agriculture as we know it?
The biggest, most powerful application by far is a strategy called marker-assisted selection, or MAS for short. This is where the tech stops being just a lab tool and starts transforming the entire process of breeding plants, making it way faster and so much more precise.
This quote really gets right to the heart of it. Instead of having to wait for a plant to grow up just to see if it has, say, disease resistance, a breeder can just take a tiny sample from a seedling and test its DNA for the marker. It's like having a crystal ball that lets you predict the future traits of a plant.
The whole process is really logical. First, you have to discover a marker that's consistently passed down with the trait you want. Then, you validate it, double-check that the link is real and strong. And finally, you use that marker to screen thousands of young plants and select only the best ones, saving a huge amount of time, space, and money.
But MAS is just one part of the story. Markers are also absolutely essential for creating the very first genetic maps of a species, a true roadmap for breeders. They're used to track down and isolate valuable genes, to confirm that genetically modified crops actually have the right traits, and even to check the purity of the seeds you might buy at your local garden center.
And this technology is definitely not standing still. The pace of advancement is just getting faster and faster, and it's all pointing toward an even more incredible future for making our crops better.
The real revolution that's happening right now is high-throughput sequencing. We have technologies like genotyping by sequencing, or GBS, that can analyze thousands of plants and find millions of SNP markers in a single experiment. This just massive increase in data, combined with a huge drop in cost, is changing the game all over again.
And all of this data is the foundation for the next frontier, precision breeding. Molecular markers are absolutely critical for genome editing tools like CRISPR. They help scientists pinpoint the exact spot in the genome they want to edit, and then they let them double-check that the change was made correctly with no surprises.
Which brings us all the way back to where we started. With this incredible power to decode and even rewrite the blueprint of life, what are we going to choose to build? Will it be crops with extra nutrition to fight hunger? Or plants that can thrive in extreme drought? The genetic tools are finally in our hands. Really, the only limit now is our imagination.